Next speaker is Regina Sloutsky from Boston University. She's going to be talking about advancing the neuroscience of post-stroke walking using task-based functional brain connectivity. I have to say I've been working with her the last few years as she's been adopting FNIRS for her studies, and I continue to be impressed by the beautiful results that she gets. So I'm looking forward to her talk.
Thank you, David, for that wonderful introduction. I'm really honored to be speaking here today, and I think that'll be better. I'm really honored to be speaking here today, and I'm really excited to share some of our recent work on functional connectivity metrics and how these might be useful for individuals post-stroke, and particularly in the context of walking. So the first thing I want to do is just acknowledge all of these people who have contributed to this work and really helped shape it, many of whom are here today, and others who are either online or elsewhere. So to start, I'll just give a brief outline of where we're going today. I'll talk a little bit about locomotor control and the effects of stroke, and then talk about how we use functional near-infrared spectroscopy and other methods to characterize walking dysfunction for these individuals. I'll talk a little bit about our specific aims and methods that we're using, and then I'll go into two different studies that we've been working on. So the first one is characterizing the neural correlates of baseline gait dysfunction for these individuals, and then look at the relationship between changes in gait and brain. So what we know about walking is that walking can range on the spectrum, locomotor control can range on the spectrum of automaticity from highly automatic to attention-demanding walking. And we see that different regions of the brain are responsible. So in automatic walking, this is predominantly driven by the spinal cord, cerebellum, basal ganglia, as well as descending drive from the motor cortex. And in contrast, we see that with attention-demanding walking, this is really predominantly driven by the prefrontal cortex, which is our thinking region of the brain responsible for executive functions. And in healthy adults, we really preserve our attention-demanding locomotor control for these novel or highly complex situations, such as this man who's taking these very deliberate, creepy steps. And so the problem with consistently relying on attention-demanding locomotor control is that there's capacity limits to what the prefrontal cortex can do, meaning there's only so much that we can think about at once. And so relying on these attention-demanding motor control strategies is problematic in the everyday world because it really precludes our ability to walk and do other things while we walk or dual task. And in fact, a primary reason why we can dual task, meaning walk while we talk and text and do other things, is because walking for typical adults is under highly automatic control. In stroke, we see disruptions in neural circuitry that happen not only at the site of the lesion, but extend beyond throughout the brain. And these result in compensatory locomotor control strategies, and so a shift towards use of these more cognitively demanding controls, such as this woman will talk about here.
Walking used to be second nature to me, and now it's like I really have to think about it.
So clinically, we hear this from patients all the time, and this increased cognitive load is a primary concern for these individuals. In addition to changes in cognitive load of walking, we see marked impairments in walking quality. And so what do we mean by that? Well, walking quality, these gait patterns emerge that are highly asymmetric, slow, and energetically effortful. And so to understand walking a bit more, we can really think about walking broadly as this series of repeated gait cycles that consist of these three major subtasks of gait. So weight acceptance, propulsion, and limb advancement. And there are lots of different ways that we can evaluate gait, but two of these ways are by looking at either variability, so this is differences across gait cycles, so from one step to another, or by looking at symmetry, and so this is looking left to right, or paretic versus non-paretic affected versus unaffected limb in stroke. And of particular relevance to stroke is the subtask of gait, which is propulsion, in which we propel our bodies forward in space. And this is measured using, one of the ways to measure this is by looking at the peak of the anteriorly directed ground reaction force. And we know that in stroke, there are these significant asymmetries that emerge as a result of impaired paretic propulsion. Propulsion deficits are in turn associated with decreased walking speed, increased energy cost of walking, and decreased community ambulation. So addressing propulsion is a primary goal of neurorehabilitation. Soft wearable robotic exosuit is an example of an intervention that targets propulsion. And as an intervention, what we know about it, the way it works is it provides mechanical assistance at the ankle, so unilaterally, to modify propulsion asymmetries. And as a gait intervention, what we know is that there are some people who respond quite well to it, and others who either have no change or detrimental effect. So we might be worsening propulsion in some of these people. So from a therapeutic standpoint, it's really important for us as a field to be better at prescribing these interventions and understanding who might be a responder versus a non-responder. As a research tool, we can kind of leverage this to use it to our advantage to start to understand the relationships between changes in gait and changes in brain. So because people have quite varied responses, we can really use that to our advantage to try to understand the underlying mechanisms between these relationships. And an important feature of the exosuit technology is that this can be quite easily turned on and off. And so when it's off, we consider this kind of a transparent mode where it really allows for normal kinematics of walking. So it's quite easy to assess what baseline walking might be, even if the device is donned. And that's as a result of it being really lightweight at the limb. And in addition to us seeing changes in, or knowing that there are changes in with propulsion and other walking metrics of particular relevance to this talk today, is that we've seen changes in exosuited induced propulsion. So improving propulsion actually leads to a reduction in prefrontal cortex activity, suggesting this shift in locomotor control strategy and potentially a reduced cognitive load of walking. And so this is some early work from our lab that's currently in review. In terms of how we can characterize walking dysfunction, so looking at both the brain correlates, neural correlates of walking dysfunction, both from a characterization point of view, from baseline dysfunction, as well as looking at the effects of interventions. So a really common way, especially in FNIRS, is to look at, if we're looking at task-based, meaning during walking, is to look at a single region of interest. And so this could be, quite often it's prefrontal cortex, but it could be other areas of the brain as well. But looking at a single region of interest and seeing how that behaves over a series of repeated trials. And this is what we did in that study that I just talked about. So this is not just us. Many groups are doing this and looking at these single regions. If we look at resting state functional connectivity, so this is often done in fMRI, but can also be done using FNIRS. And so the idea behind resting state is that it's task agnostic, but it does have some value in terms of predicting motor outcomes and also evaluating changes of interventions if we expect lasting changes. So over a period of weeks or multiple sessions, if we expect to see changes in the brain, we could potentially see these using resting state fMRI. But fMRI is fundamentally not conducive to walking, right? Because you have to be lying very still, and so the best you can do is imagined walking or maybe simulated walking, but definitely not real life walking. And so if we think about moving towards how we can evaluate task-based, use functional connectivity for task-based evaluations, this is a really nice quote from this paper in 2011 that talks about how a connectivity-based perspective really seems to be much closer to the neurobiology underlying brain function compared to approaches that assign specific clinical symptoms to anatomically segregated regions. So we know the brain is really complex, and looking at it in a single isolated region might not make sense from the physiology of what's really happening in the brain. And in addition, there's a recent consensus statement that was put out by leading experts in stroke recovery that talks about the importance of looking at task-based connectivity to predict treatment response across stages of stroke recovery. So we know that this is important, and we want to try to find ways to achieve this. I'll talk a little bit about different functional brain networks that are involved in walking, and so I'll introduce you to each of these that we're going to be evaluating, and then I'll go into looking at talking about how the interactions between these might impact walking. So the three networks that we'll talk about today are the default mode network, the frontoparietal control network, and the dorsal attention network. The default mode network is primarily active at rest and during mind wandering, so it's our kind of spontaneous thought network, but it also can be engaged during intrinsically directed cognitive tasks, so introspective thinking, things like autobiographical, self-reflection, thinking about interpersonal relationships, things like that. And then we have our two attentional networks. So these networks are active primarily as during a goal-directed task. And they differ a little bit in the sense that the frontoparietal control network is our cognitive attention network and responsible for things like executive functioning, planning, problem solving, and organization. And in contrast, our dorsal attention network is our visuospatial attention network, and it's responsible for taking in sensory information and then coordinating motor processing, so things like planning a route of how you might walk somewhere. And so when we think about what functional connectivity is, it's really this correlation between networks. And so there are different ways of looking at it, but what we're looking at today, the way that we're thinking about it is just a correlation between activity between each of these two networks. And so we'll explore the three different comparisons that we have here. The first comparison is looking at default mode to dorsal attention network. And so here we're characterizing neural flexibility. And so we know that a hallmark of the healthy brain is anti-correlation between these two networks. And so we can think about it as flexible recruitment or anti-correlation, where one network is suppressed as the other is upregulated, really allows for the interplay between these networks really allows for flexible switching between internally and externally focused cognition. So here, flexible recruitment would be complete anti-correlation, and inflexible recruitment of the two networks would be positive correlation of 1. The next networks that we're going to look at, so the frontoparietal control network as it pairs with the dorsal attention network, and so we can think about this as characterizing walking automaticity. Synchrony between these two networks suggests that there might be increased attention to the motor task. And so if your two attentional networks, or two of them, are paying attention to the same task, we think that they might be both needed at that moment to pay attention to how you're walking or moving. On the opposite end of the spectrum, we think about if you're able to engage one of your attentional networks for thinking as the other one is paying attention to walking. This is more of an automatic control strategy, more automatic walking. And then the third comparison here is looking at the frontoparietal control network to the default mode network. And here we're characterizing a focus of cognition. And so if these two networks are paired and synchronized, their activity is synchronized, then this suggests a more internal focus of attention, so more of that introspective thought or intrinsic cognition relative to more extrinsic cognition. And we know that there's a relationship between resting state fMRI and looking at the dorsal attention to default mode networks, the connectivity between them, and gait impairments. And so this study looked at 3 different clinical populations. And what they found is that what you see on the y-axis is gait variability, and so this is one of those measures of gait impairment where a greater value suggests more variability and worse walking. So here what we're seeing is that the people who are anti-correlated between these two networks actually have better gait compared to the people who are more correlated. And so this suggests that there's this relationship there between functional connectivity and gait. And some of our work, what we've done is we looked at using FNIRS to evaluate functional connectivity between these two networks. And so this was not resting state. This was during standing, a period of 2 minutes. And what we saw is that same relationship where worse walking was associated with more correlation between the networks. And so what we wanted to do in our work is to really evaluate how we could use task-based functional connectivity to look at the clinical utility of using task-based FNIRS-derived functional connectivity metrics during post-stroke walking. So in the first study, we looked at characterizing baseline dysfunction, and in the second study, we looked at the relationship between exosuit-induced changes in functional connectivity and changes in propulsion symmetry during performance of a real-world functional task. This was our FNIRS probe, and so we used 32 sources and 32 detectors, and these were the regions that they covered, and of course these are the cortical projections of these networks. Because FNIRS is limited to the more superficial cortical measurements, these are the channels that we used for each of our networks to define them. And what we did is we had people walk in this real-world complex environment, which was their university garage here at BU. And they walked for over 30 seconds, but we only analyzed the first 30 seconds of walking for this task. And so the first thing that we looked at was baseline characterization. So this was with an unpowered exosuit. And we had 20 participants do this part of the study. And then what we had them do is perform this dual task. And so they did it in a randomized order, once with an exosuit and once without. And the dual task was trying to find a car in an unknown location in the garage. And so we gave them a key fob and we asked them to walk around and use really any strategy that they wanted to try to find the car, but we really encouraged them to use that key fob to try to find it. And here you'll see, and maybe here, I don't know if anyone could hear that, but he was pressing the key fob, and you could hear the kind of the car beep, car alarm going off, and so he's looking for the car. So this is a highly uncontrolled environment with cars driving by, people, anything that you might see in a regular garage. And for this part of the analysis, we had 13 people complete this part of the study. So first we'll look at that baseline walking and characterizing a baseline gait dysfunction. So when we looked at the relationship between the dorsal attention network and the default mode network, we saw that same relationship during walking. We did not see this, we looked at it in standing, we did not see the same relationship in standing, but when we looked at it during walking, here we're looking at propulsion symmetry. 50% is perfect symmetry, and so that's less impairment. So you'll see the line is going in the opposite direction, but the relationship between impaired gait and connectivity is the same. So people who have more of that flexible recruitment seem to be walking better. If we split them into subgroups, so now we look at positive and negative, flexible and inflexible, we see this relationship start to emerge where we can try to categorize these people by how their brain activity is, characterize them by their functional connectivity profile. And so we see these significant differences between people who are flexible versus inflexible. And subgrouping these people might have clinical value down the line in terms of predicting the response to intervention and predicting who might be a responder based on their underlying mechanism of what might be causing their asymmetries and their gait dysfunction. So the next thing we did was we looked at our two task-based attentional networks, and what we found is that there was a relationship when we looked at this cognitive focus, so the frontoparietal control to default mode network, but the walking automaticity network in and of itself was not an independent predictor. And when we put these into a model with the three networks, what we saw is that there was a significantly improved R-squared values, whereas each of the two significant independent predictors had a 22 or 24% R-squared. All three of these together in a model went up to 50%. And so we think that looking at all of these might be important. The next thing that we did was we looked at interactions between our neural flexibility network and each of these two attentional networks. And so what we found is that there was no significant interaction between the cognitive focus network, but there was a significant interaction between walking automaticity and default mode or neural flexibility. And so when we put these together, we saw an increase, further increase in an R-squared. So this model explaining 61% of the variance. And so what does this look like? So essentially, when we put it together, we see that people who are flexible, it doesn't really matter if they're using a controlled or automatic strategy. They seem to be doing quite well. On the other end of the spectrum, if we look at people who have this inflexible recruitment, people who are using a more of an automatic control strategy might actually be walking into their impairment and exaggerating their impairment, whereas people who are using a controlled strategy might be using that as a way to compensate for this reduced flexibility in their neural recruitment. And so if we look now at combining our metrics of automaticity, our scale of automaticity and flexibility, we can start to think about 4 different subgroups. And so these 4 different subgroups emerge where we see significant differences between people who are inflexible and people who are flexible. And so these more automatic these inflexible automatic individuals seem to have lower propulsion. So this might explain some of that, some of those underlying mechanistic differences in what's causing their asymmetries. So key takeaways from this study, we think that we might be able to use task-based functional connectivity using FNIRS to characterize propulsion symmetry during walking, and this may help us understand the underlying mechanisms of post-stroke gait dysfunction. And so there's potential for this clinical utility in prescribing targeted, individualized gait restorative interventions, and that's a little bit about what we'll explore in the next study. So in this study, we were looking at the garage car finding task. And so here we're looking at the first thing we wanted to do is kind of validate our task. And so what we did was we looked at single task walking compared to dual task walking in the attentional network that had its own significant response that was individually significant. So this is that frontoparietal control to default mode network connectivity. And what we saw is that consistent with what we would expect, as you transition into more dual task walking, we see a shift towards more of that extrinsic focus of attention. And so you're paying more attention to what's going on in your environment and to the motor task relative to when you're just walking. And interestingly, what we see is that here, the people who are highlighted in red, those are the people that had the reduction. So the majority of people really had a significant reduction here in that dual task. So this just suggests that we were able to capture the complexity of the task using our paradigm. And then when we looked at the relationship between changes in connectivity and changes in symmetry using during exosuit versus exosuit assisted versus exosuit unassisted walking, in that same network, what we saw is that changes in propulsion, so an improvement in propulsion was associated with more change in the more intrinsic walking cognitive focus. And so this is exciting because we can start to think about how we can evaluate effects of interventions in real time in a single trial and look at these interventions in terms of how they're acting on the brain. And so if we look here, we weren't really powered with our 13 people to do statistical analysis, but if you look here, we can start to think about how we can prescribe these different interventions and the variability in response. And these subgroupings might not necessarily be the best subgroupings to use or the most correct ones to use. But if we start to think about how people might respond in these different subgroups, we can maybe try to think about bringing in brain connectivity as a way to understand how people are responding or not responding to these different interventions and come up with the best approach for them. So key takeaways from here, functional connectivity metrics may be useful for characterizing task demands, so looking at dual versus single task walking. And then changes in functional connectivity metrics are in this particular network are associated with changes in propulsion. We still have some work to do in figuring out the clinical utility of looking at the immediate effects of these gait restorative interventions, but this is an important step in that direction. In general, we think that looking at multiple networks and not just focusing on single regions of interest are really important to think about when we try to understand more holistically how the brain is working during walking. So we know it's complex, and looking at it in a more complex, comprehensive way is important. We think that there's value to using these functional connectivity metrics for evaluating response to interventions. But we recognize that we really need a larger sample, and so using a larger sample here might help us identify better cutoffs, inform more of the prognostic value of using functional connectivity subgroups to predict treatment response. And then in terms of future directions, we might want to consider extended connectivity analyses, so looking at different cognitive or attentional networks or interhemispheric connectivity, which is of particular relevance to stroke, looking at within-network connectivity, looking at effective connectivity to help us understand causality, and then looking at more coverage. So if you notice, we had very few regions, very few channels characterizing each of these networks, but potentially higher density arrays might help us. And then it was really exciting to see all of this in a garage, real world, really complex, really uncontrolled setting. But it'd be, I think, important to take a step back and see how these relationships are in a more controlled setting where we can really understand what's happening a bit more. Thank you, everyone. Thank you to all of you and to our funding sources. And I'm happy to take any questions. Sorry for going a little over.
Hi, very wonderful talk. I know how hard to do the motor study, use the FNIRS. So I have two questions. The first one about the Atlas, as I guess correctly, you might use the AAL Atlas. So I was wondering why, since it's a task-based analysis, why you don't use the functional defined network? And if you did, have you compared the difference between the AAL and the other Atlas or not?
I'm sorry, you're saying the atlas for like how we defined our regions or how we define our networks?
Yeah, how you define the networks.
Yeah, so great question. So we looked at the cortical representations of these networks, and so we're not always able to get at the deeper structures, and we're not always able to get to everywhere we want to be. And with limited number of optodes, that's really, we're playing around with kind of optimizing our optode arrangement for what we can get. But we picked regions that we thought were specifically in each network. And so if you look at if I go back to the brains that show where the regions are, maybe it's too far back, but we try to not have any overlap in where we, and so any overlap in the regions that we're assigning to each network. Does that make sense, or does that answer your question? Or no?
I think I'm rewording my question. So as I saw, you have the definition for each network, like a default network and also light attention network. So that's, I think, is special structure-based atlas is from AAL atlas, is correct?
The AAL, yeah. So the definitions, are you talking about the definitions of what the networks do, or the definitions of where we're measuring the networks?
I think in FMR or MR literature, they have special structure-defined network is most famous one
Okay.
is
So,
AL,
yeah.
and then they also have functional defined network like peso, golden peso.
Yeah.
Pacylation or the power pacylation. So my question is why you use the structure-defined network instead of the functionally defined network?
Yeah, so we actually did try to use the functionally defined networks. Yeah, so sorry, I didn't quite understand your question, but yeah, these were functionally defined networks, and so there are corresponding brain regions to the functionally defined networks, if that makes sense.
Okay, thank you. And the other question is about the functional connectivity. Again, in the FMR literature, they suggest use at least 20 minutes to do the connectivity analysis. So I was wondering how long do you use to do your analysis, and what's your suggestion about the time period?
Yeah, so we did 30 seconds for our analysis here, and some of that was driven by how long these people were walking, and so we wanted to capture the amount of time that they really were looking for the car. So after they found the car, we could think of that as not really the same as like using the key fob to find it. So if you once you know where you're going, that's a little bit of a different task. So we really limited them to those first 30 seconds to just try to capture that moment. But we didn't play around with any of the different time periods. So, I don't know if maybe using a shorter or longer time period would have led to different results, but that's an interesting direction to take this in to really start to understand what the minimum amount of time is and what the minimum amount of time is and whether we can start to understand what happens over time a bit better.
All right.
But yeah, thank you so much.
Thank you for your presentation. I have one question. How did you handle the motion and systemic artifacts in the functional connectivity analysis?
Yeah, so we used motion correction algorithms. Obviously, we expect them to be moving their head. They're looking around for a car, so there's no really getting around that. And so we did the best we could with correcting. We used spline and wavelet motion correction algorithms. And kind of visually inspected the data to make sure that there were no huge motion artifacts beyond what we would typically expect in regular head motion. So that's what we did for that. And then we also used short separation regression to account for superficial regions of the brain and extracerebral activity. So yeah.
Thank you.
I'm sorry to press you on this, but you must have had challenges. Speak something
Oh,
about
yeah.
the challenges.
Oh yeah, there were challenges. I make it. I can talk day and night about the challenges. Any particular challenges you want me?
Briefly.
Yeah. Challenges with motion and specifically, or just in general? I would say, so we had some challenges with designing the probe, so starting from
The motion.
The motion.
Because that's a big issue with the
Yeah. I wonder if potentially, because we had so much motion, that it was really just, it was so uncontrolled, and we really expected this, we expected head movements, right? And so we weren't telling people you can't turn your head as you walk around a garage, because that would be unsafe. And so, visually inspecting the data was helpful, but I really didn't need to do a whole lot of pruning of the channels, or there were no condition, like I didn't drop anybody because of motion.
So cap design must make it more stable.
Cap design and bald heads.
Got it. Good. Next.
Hi, that's a short question. Apart from the task, I was thinking about the importance of sensory afferent that are involved in postural control. And my question was that, for example, if we compromise someone's vision or simply ask them to close their eyes, okay, I think we are decreasing the correlation in that spectrum, correlation between the dorsal and frontoparietal attention. But this reduction in the correlation is necessary make it more controlled or more automatic. Because I think
So,
Doesn't make it more automatic, but we are still decreasing the correlation.
so if I understand you correctly, you're saying if somebody closes their eyes, I would say that they're still planning their path, right? And if you close your eyes, you probably actually have to plan your path even better, right? So if I were to close my eyes and have to navigate through this room, I would probably need to be very careful, and I would probably not be using an automatic strategy as my hypothesis. I don't know for sure, but I probably wouldn't be like just casually walking, and because I'd be
It's
afraid
more
to.
controlled, yeah?
It's more controlled, yeah.
Based on the spectrum you showed, the correlation is shifting toward negative one, so shouldn't it be more automated?
I don't know where that slide is, so if it's a.
Too far back, sorry.
Am I going in the right direction? Yeah, I think I am.
Here.
Yeah. So if they're working together, so if they're both paying attention to the same task, we think about that as more controlled walking, because you're thinking about your walking in terms of kind of your frontoparietal control network, your dorsolateral prefrontal cortex is thinking about it, and so you're like, gotta not, run into something, and also you're planning your path, and so you're working together towards using more of that. It's a common goal, if that makes sense.
Thank you.
Yeah, thanks.
Hi, thank you for this great talk. So I'm curious about this relationship between this neural flexibility, if that's the correct term, and gait. So I was wondering, this neural flexibility, to what extent do you think this is a more like an interpersonal difference that kind of pre-exists in healthy people, or whether this itself, like a reduced neural flexibility in itself, could be an effect of stroke? Mm-hmm. And the second part is, to what extent, say, if you have a measure of this neural flexibility before stroke, could this be useful as a predictor of say, recovery, success and progress?
Yeah, really interesting question. So we see this neural flexibility, and whether this is the correct terminology for it, but basically this anti-correlation, we see it across all sorts of different clinical populations, ranging from ADHD to schizophrenia, depression, really across the board. And so we think that it's really a hallmark of the healthy brain to have this neural flexibility. But I don't know if... so there's a lot of different confounding factors, right? Because you might it might predict your recovery to stroke, but we'd have to measure it ahead of time and then see whether or not you have any of these other conditions, and potentially, it's also maybe impaired. And I think it's also impaired in sleep. It's impaired in all sorts of different clinical cohorts. But it'd be really interesting to see whether that could be the case if we could start to do these connectivity analyses for everybody and get bigger data sets. And your second question, or did I kind of combine the two a little bit?
Yeah, I think the second was just if it's more like an interpersonal difference, could we use, if we measure that beforehand, could we use that to predict? But you answered, I guess.
And one other thing to your question, one other point is, so we think that the exosuit, for instance, might be affecting these more of the attentional networks, but there might be other interventions that could affect the neural flexibility that we just haven't really explored. And so that maybe there's the combination of enhancing flexibility and something like an exosuit that takes on some of the control of walking that might help. Yeah.
Yeah, really interesting. Thank you.
Thank you.
Okay, one more question.
Thank you. Hi, I'm interested in your gait measures. You chose gait variability of stride time, it looked like, in terms of your good walking versus your bad walking. And I was just wondering if you've done any balance metrics. Sometimes we think with pathology we actually get a reduction in variability as opposed to expansion of variability. So just interested in whether you've thought about that or looked at those.
Yeah, thanks for your question. It wasn't, we really, the standing analyses where we looked at gait variability weren't really a primary focus of this task-based work. And so we haven't looked at any of the balance metrics. And what we know in stroke is that variability is typically increased, particularly stride time variability. And so we didn't look at different variability as it relates to propulsion because we had this propulsion modifying device. And so really we were focused on kind of the propulsion problem, but it'd be really interesting to look at these different variability metrics or even just different gait metrics in general and see, different interventions target different things. So that'd be an interesting future path.
Great, thank you.
Thank you.
Okay, thank you very much, Regina. Great discussion again.